Hello, and welcome to the second webinar of our Let's Talk Disease series, presented by the Ontario Ministry of Agriculture, Food and Rural Affairs and Greenhouse Canada magazine. Each webinar in this series will be moderated by an OMAFRA greenhouse specialist. Today, we have Dr. Sarah Jandricic, who is the Greenhouse Floriculture IPM specialist in Ontario. Sarah has over 18 years of experience in greenhouse IPM, including extensive research experience in Canada and the US, and 2 years experience in greenhouse IPM consulting. Responsible for both IPM of insects and plant diseases, she has authored a number of trade articles and runs a variety of grower workshops related to the biological control of greenhouse pests. Welcome, Sarah.
Hi, Greta. Thanks so much. So our webinar today is going to be from Dr. Ann Chase talking about Fusarium. And for those of you who aren't familiar with Ann Chase, she's really one of our premier experts in North America on floriculture plant pathology. So we're really lucky to have her here today to share some of her expertise, and there'll also be a chance to ask questions to her at the end as well. So that's a really valuable opportunity. So Dr. Ann Chase has close to 40 years experience in research, diagnostics, and practical consulting in plant pathology. She was previously a professor of plant pathology at the University of Florida at the Mid-Florida Research and Education Center in Apopka. And Ann has remained on staff as a professor emeritus since her retirement in 1994. She continues to provide practical solutions for growers through Chase Agricultural Consulting. So before Ann gets on, I just want to go through a few housekeeping things for everyone. So you should all see a dashboard panel on your right with options in gray. Ann's been nice enough to provide handouts of her slides. So if you see the little handout tab, there's a little arrow, a triangle beside it. If you toggle on that, you'll see a link to a PDF of her handouts. So you can click on that and it'll open in a separate screen. And then you can either print those off to take notes today, or you can use that to save later. Also, this webinar is being recorded, so if you miss anything that Ann says, you can definitely go back and listen to it afterwards. And the last part is the questions, the very important questions tab. Again, if you toggle to the little triangle beside the questions word in gray, that'll open up a box for you to be able to type your questions into. And at the end of the talk, I'll go through some of those questions and ask them to Ann, and we'll get her opinion on things. So with that, I'm going to hand it off to Ann, and this is going to be really exciting, everyone.
Thank you very much, Sarah. It's interesting. A long time ago, right before I left University of Florida, I actually was introduced as comic relief. So I think it's probably a little better to be introduced as exciting. At any rate, I do appreciate the opportunity you presented me to give the audience a review or an overview of Fusarium. It's one of the toughest pathogens to control that we have, probably in some respects I'd say the toughest. And after we go through the presentation today, maybe you'll see some of the reasons why you might be struggling so much with it. I'm going to go ahead and cover it in a variety of sections. But before I say anything else, this is our newest greenhouse that we have here in Arizona. And having gotten extremely sick of airplanes, I'm now doing trials again. So I'm having a lot of fun finding some new information out about controlling diseases on ornamentals and other greenhouse crops. So what we're going to talk about, I'll start off giving you an overview of the symptoms that Fusarium can cause, and there's an amazing array of them. We're going to cover the host of Fusarium, so more or less the most common host that you might find in ornamentals. What Fusarium's like as a pathogen, why is it such a challenge? What are its characteristics? The importance of sanitation, both of surfaces like benches and pots, and then finally a little bit of sanitation work on plant material like bulbs. The importance of Fusarium control using fertility is that I can't stress it too much, and it's actually truly the most important thing for you to remember from this webinar, in my opinion. I'll show you some of the trials that have been conducted in the last few years on Fusarium, including biological and conventional products. and they are not very many. We don't have a lot of people even in the U.S. who work on ornamentals, but I've gathered them together as best as possible to give you an idea of how I come to the conclusions I do about what will work and what doesn't work very well, or at least not consistently. And then finally, I'll give you a summary of the very best products from biologicals to the conventional. And Sarah was kind enough to provide me with a relatively current, at least, list of what's registered in Canada. So I've tried to be cognizant of the fact that you don't have as many products as we do in the U.S. for disease control, whether they're biological or conventional, but we'll go over those at the very end. So to start off, many of us think of Fusarium as being a Fusarium wilt situation. You see here a typical Fusarium wilt on chrysanthemums. This happens in the U.S. at least every year, as many of our plants are grown outside in full sun, and they do get the full brunt of the environment. It starts off causing a one-sided type wilt that you might notice here in this upper left-hand side, graduates to much more severe symptoms, and you eventually get to the point where if you cut into the stems, you can see vascular browning, this picture at the bottom right. In many situations, though, too, you actually see the signs of Fusarium in this mycelium, or the mat that the organism causes. This is very often white and can be pinkish or a pale orange. And that's one of the things that I look for when I'm trying to figure out if I'm looking at a Fusarium disease or something else. But it really doesn't stop there. Fusarium is able to cause leaf spots. Upper left corner, this is actually a Fusarium stem rot, leaf spot, depending on how you refer to Christmas cacti, but it is an aerial disease. And you see that, and you can see the sporulation here in these little pustules. We also find that Fusarium, at least on some tropicals, will cause leaf spots. The mother-in-law tongue or Sansevieria snake plant, these are caused by Fusarium. They're relatively unusual for what Fusarium can do. If we go to the upper right-hand corner, these little speckles all through here on this philodendron are also Fusarium leaf spot. And finally, in conifers, pitch canker on pine can be devastating disease, another aerial or foliar disease. Other symptoms, very often we will see stem rot. You see here on this plant, just a clear-cut stem rot. It could be caused by a lot of different things, but it happens to be Fusarium here. If we move over to the root systems here, this Coreopsis has Fusarium on the right, whereas the healthy one on the left, showing just standard root rot symptoms. There is no way for you to look at the roots of a plant and tell which pathogen is causing a disease. All you can really tell is that you have a pathogen. The bottom slide shows one of the most common diseases on many types of dianthus, which is a crown rot. It's very common on with Fusarium in this case. And then finally, in some cases, all you'll see is a major stunting of the plant. The healthy plant on the right side here versus a stunted one, and this again is Fusarium. Getting down to another one that you might be more familiar with or consider common for Fusarium is bulb rot. In some cases, like on this Crocosmia, we see a vascular infection. This is Fusarium wilt. If you look at calla lilies, bottom bulb that's been cut here, it's not a vascular disease, and you see these little spots of kind of confined rot, a dry rot type situation. This is really common on calla lilies, but not on some of these other plants like, say, tulips or hyacinth. They very much more often have a vascular wilt. And then finally, probably the plant in ornamentals that gets the most research when you start talking about Fusarium, and that's cyclamen wilt. This is a true vascular wilt on this plant. You can see if you cut through the corms that you see the rot that's surrounding the vascular system. And then the plants on the bottom just show you the range of symptoms that can be caused. If a plant has Fusarium wilt, it will die. There's nothing you can do about it. If it has a stem rot or this kind of bulb rot, like on the calla lilies, there are some things you can do, and sometimes you can even outgrow the disease by good cultural conditions. finally, I wanted to show you a few more pictures of what Fusarium will do, and it's just extremely happy, and it's growing all over the surface of the plant. If we look in the upper right-hand corner, you see again all that masses of mycelium growing there. There will be a little sporulation as well. And again, to my eye, it is not a pure white. It has a little pinkish tinge or sometimes a pale orange tinge. Same kind of thing on this tree stem, thoroughly dead, but we see the Fusarium popping out all over the place. And if indeed you put it in a culture dish as a pure culture, it is the same color. And the way that it grows is the same. So a lot of times if you have experience in doing diagnostics, you can get a hint of what's causing the problem. The one at the bottom, remember I showed you that picture of leaf spots on mother-in-law tongue? This is the Fusarium that causes that. This one's actually lavender. And some of the cultures that we see on certain Fusariums will be a very distinctive purple color. So again, when you're looking at a rotted plant, sometimes we're looking at color differences to help us with a clue about what might be causing the damage. So I've shown you some of the things that are Fusarium hosts, but the list is just amazing. Almost all types of bulbs are a problem if they are contaminated with Fusarium, from tulips to daffodils to gladiolus. Just about all of them have a big problem with it if they are infected when you get them. Another group is many tropical plants, whether we're getting leaf spots or crown rots. Tropical plants seem to be a favorite host for the Fusarium pathogens. Herbaceous perennials like hostas tend to get Fusarium pretty regularly. Cut flowers, another problem child. I mentioned the bulbs before with gladiolus, but they are a cut flower in many instances. That's a problem for them and many of the others, like carnations. Bedding plants, especially dianthus. Flowering crops, like the mums we looked at the very beginning. And finally, honestly, just about anything in propagation, whether it's a woody plant or not. So this list pretty well covers ornamentals. There's just about nothing that escapes it. You don't see a whole lot of Fusarium on turf grass, but then they aren't really considered ornamentals either. So the range of hosts can be amazingly diverse, and it does include just about everything we grow in ornamentals. So the characteristics that I wanted to highlight, this is what makes this organism, this fungus, such a challenge to control. In the first place, it makes a lot of spores, and they're really easy to spread with water, which means you can splash them with overhead irrigation or with a hose. Rainfall can splash them. They can move in an ebb and flood system really well, so they're really easy to spread by water. They do, as I showed you with the symptoms, affect every part of the plant, from the leaves, the crowns, bulbs. They cause wilt, and they cause root rot. And they don't do all of these things on all plants because there's some specialization, but they can affect all parts of the plant. Some of the species of Fusarium are really wide in their host range, so that if you had a disease causing damping off in a plug range and you grew tremendous number of different ones, you might find that it spread to a lot of them. In other cases, it'll be very narrow. The Fusarium that attacks gladiolus does not attack anything but closely related gladiolus plants. They can be good saprophytes, and this is probably the biggest problem with trying to control Fusarium. And by a saprophyte, it simply means an organism that does not need another organism to live. So it would be like something that just lives off of the decaying debris or off of the exudates from a plant and not attacking the plant necessarily. They can attack plants, but they don't have to. So as good saprophytes, it makes them a tough thing to control because they have more than one way to go about their life. The Fusarium diseases do thankfully react rather dramatically to the form of nitrogen that you use. So we'll go over that in a lot more detail later. But this is the most important point. As I said, the form you use will dictate whether or not you can control a Fusarium disease. They also react to plant stresses. So if you cause stress to the plant by either heat or lack of water or too much water, you can kick off Fusarium. Years ago, when I first started working with the Fusarium wilt on cyclamen, Marjorie Daughtry told me that if I wanted to make it happen, I needed to let the plants wilt. And she was right, absolutely right. You would think, as with pythium, that too much water would be what triggered it. That's not the case with Fusarium. It's very often stress, whether it's lack of water or flowering. And as plants go into flowering, they're changing their physiology, and that is a real trigger for vascular wilts to start causing a big problem on that plant. Some of them are spread by insects. And this is unfortunate, isn't it? Now, that gives you one more thing to have to watch. I'll show you some examples of research that was done on spreading Fusarium on Lisianthus. And finally, probably one of the worst things, if you are actually growing in the field or outside, is that even when the host plant isn't there, this kind of organism can stay there for years and even reproduce on the roots of weeds and other crops. So that goes back to the point about it being a tremendously effective saprophyte. Amazing, amazing adaptive organism. So let's start off and talk a little bit about waterborne pests. What makes something a waterborne pest? Not just something that's in the water, but what makes it a problem? So it's a pest is a problem, basically. Anything that makes a lot of spores very fast can be spread by water well. It's just something that helps make it an issue. If they have motile spores or ways to swim, algae do, and the bacteria do, Phytophthora, and pythium can make spores that actually can swim, and that is a serious problem, obviously, in a waterborne system. As we talked about earlier, if they're good saprophytes, that's much more likely to make them a problem in water. If they either are water-loving, like pythium or Phytophthora, or at least tolerant, then that makes it more of an issue. If they make long-lasting resting spores, and if you think about how plants, once they're finishing their life cycle, they make a seed, that's in essence a resting spore for the plant. So fungi and bacteria actually make types of structures that will last quite a long time too. In the case of Fusarium, they're called chlamydospores. If they have a wide host range, and we talked about that, that means that there's very little you can do if you just simply get rid of the plant that you thought they were on. And then finally, if they have complex ecosystems, meaning they interact a lot with other organisms, it's a little bit like thinking of the bioslime that's in our irrigation tubes, that is a combination of a great many organisms that create that little ecosystem there. So what's Fusarium like? This is what I'm showing you right here is just what waterborne pests, what their characteristics are. Here's the characteristics of Fusarium looking at that same list. Everything that's in the orange is true of Fusarium. So it does not make motile spores. It can't swim on its own, but it's moved by water pretty well. And it's not water loving, but it's absolutely water tolerant. So this is why Fusarium is an issue with water movement. Now, the reason you want to know this is if you want to prevent spread to another greenhouse or spread within your own greenhouse, if you're recirculating water, then you need to realize that it is going to be something you should keep in mind. We're actually working right now with a grower, a Gerber daisy grower in Southern California, who is using the standard method for cut flowers and recycling water, and going to a great deal of trouble to treat water because the Fusarium has cost them a tremendous amount of money in losses and replanting the crop. So Fusarium definitely moves very well with irrigation water. I just thought for the heck of it, I'd show you a few of the spores. Fusarium is known to make two different kinds of spores. One is shaped like these, kind of pointed ends. These are called macro spores, and the other ones are called micro. And that's these little round guys. Not round, I'm sorry. They're ellipses, oblong. Maybe that would be the right word. At any rate, because they make the two spores, it just has them more adaptive. It doesn't, I don't think anybody's ever done any studies that I'm aware of that shows that one or the other is more important. But the thing is see how they're kind of sticking together here in these microconidia? They make a sticky substance. That makes it also harder to kill them because they're a little bit protected, and it does make them stick well to surfaces. So it's something that you have to keep in mind, that they're not just going to wash off all by themselves. So the next section I want to talk to you about a little bit is, what do you do about surfaces in between crops? If you've had a crop that had Fusarium, what do you do to prepare the bench or the floor or the ebb and flood system, whatever it may be, so that you don't bring the disease along into the next crop? Now, we did these trials when we had our business in Northern California, and we actually looked at polyethylene, film, concrete block, and wood. And when we did the trials, we made sure we were using old surfaces, so nothing that was brand new and would be easier to clean. As you can see here, we actually were growing on wooden benches, and we had no trouble growing algae, just like every other grower that starts to produce plants in a greenhouse. So we're using these three surfaces, and what we primarily were looking at that point was trying to kill Fusarium oxysporum or Phalaenopsis vesicola. So because you were talking about Fusarium today, I thought I would show you that. The examples are comparing several different disinfectants and a cleaner, and it just shows the difference between the percent kill. So ideally, what we want to see is not a zero, because what this says is that when we treated the plastic surface with just water, we didn't kill any of them, which one would hope you wouldn't kill a whole lot with water. If we treated it with Strip It alone, we killed 72% of them. So Strip It is a soap acid type product, and it is very bad on plants, but just great on cleaning surfaces. The next product we were looking at was Xylitol. In this case, the Xylitol on the plastic by itself killed 75% of the Fusarium. But when we pre-treated with Strip It and then followed with Xylitol, the kill was, basically there was a bad interaction apparently between these two chemicals. So that was a pretty negative response. Going to Green Shield, which is a third generation quaternary ammonium product. Green Shield by itself or with Strip It gave us about with the high 60s in percent control. So there was no benefit really to adding the Strip It to the Green Shield. But the percent control, if we just jump right to this last column, the percent control was 100% kill if we started with Strip It and then followed with Clean Grow. Clean Grow is a product from a company in Vancouver called Pace. It has a different registration actually in Canada than it does in the US, so it's kind of hard for me to keep up with which things are registered where. But Clean Grow is a quaternary ammonium, but it is a fourth generation, so it has different characteristics than any other quaternary ammonium on the market. The program as a whole says that they have promoted is use Strip It first to clean the surface. You leave it on for 5 minutes, rinse it off, and then follow with Clean Grow and leave it on the surface. So that is what we were testing here with these other products to see how it stacked up. I've just shown you one of the trials. I'll show you a summary of those trials across the three different surfaces. So you see, again, we looked at just water by itself, Clean Grow, the DDAC, Green Shield, the third generation quat, and Xylitol. If we look just at the wood surfaces, wood alone, the water by itself didn't do anything, which again, really hoping we didn't kill stuff with just water. Then we had 35% control with Strip It on wood. If you look at the Clean Grow, it did not work well on the wood alone, but with the Strip It, we've got 100%. So we've got an interaction, and that's not really an interaction. I shouldn't have said it that way. It's more of an additive benefit of using the cleaner, the Strip It first, and then that. And then looking at Green Shield, you can see much less results, and Xylitol was really ineffective for Fusarium on wood. Look at the plastic surfaces, it's similar, but again, the best control that we're seeing is with Clean Grow and Strip It. The Green Shield did just as well in this plastic surface. As you can imagine, plastic is quite slick. It's much easier to contact everything on plastic than it is on wood. And then the xylitol did 100% effective on the wood with the strip at first, and then nothing by itself. And then the last column is the concrete. Again, at this point, we're seeing the strip at 100% effective on concrete all by itself. But again, that additive benefit of adding the two, or not adding them together, of course, but using that program. So in the experience that I've had over the years, what we end up recommending for cleaning surfaces is the PACE program. Move off this a little bit and give you an interesting result that we heard from Janice Elmhurst, who is she's just now retiring, as I understand it, plant pathologist in BC. She did a lot of trials over the years. In this particular one, she was looking at cyclamen plug production. So these tiny little plants, as you know, any kind of slow-growing plant, algae and moss take over really fast. So in this particular case, she was primarily looking at trying to control algae in the plug production. What she ended up finding out, in addition, is that she got control of some diseases. But if I first just show you the algae, here is clean grow at a low rate, 1/4 of an ounce per gallon, and I can't off the top of my head flip that into metric, but hopefully we'll be able to do that later. And then the high rate, what you can see is dramatic reduction in the algae. A rating of 10 here is 100% covered with algae that the water had. And then as she used a little bit of clean grow in that 14-day interval, so one treatment every 14 days, we've got significant and really dramatic reduction. A rating of 1 here is no growth whatsoever. After 4 weeks, we can see it going down even more. And finally, at the 5-week rating, the high rate was the very best. We had virtually no algae growing on those flats. But we're talking about Fusarium. So this treatment, this one right here, the 0.25 ounces per gallon, gave 94 to 100% control of Fusarium and botrytis on cyclamen seedlings. So compared to the water untreated ones. So this treatment did do more than just control algae. And if you start thinking about trying to control diseases on plants, we have worked a tremendous amount with Clean Grow to look at what diseases it will control and which ones it's not as good on. But in this case, Janice got some tremendously good results with the cyclamen plug flats. So if we switch off again from the plug flats, she also did bulb dips. I have been 100% against dipping anything for control of diseases, and finally had to give up because I saw too much data, and I'll show you all that right now. That makes me say that I do think bulb dips can be very helpful. Janice's work, she split it up into daffodils and tulips. So I'm going to show you the daffodil data first. These were field grown in Canada. They were naturally infected with the Fusarium, so it wasn't anything that was done in a lab. And she then grew the plants out after different treatments. The Clean Grow rates for the dip, it was a 5-minute dip only, no rinse, just a 5-minute dip. And what we see here is from 71% survival of the daffodils when they were not dipped to 95% if they were dipped. The number with Fusarium dramatically dropped. The height was different as well. And honestly, if you just focus on the Fusarium data, the dipping was really important on the daffodils for knocking out the plants with Fusarium and for allowing them, many more of them, to grow and become saleable plants. If we switch over and take a look at the tulip data, in this case, she used 2 different kinds of tulips. One of the cultivars was really susceptible to Fusarium, and one of them was really resistant. In this particular trial, what she found out was that, again, that dip dramatically reduced the number of diseased plants a lot less in the dipped tulips in this one cultivar that was susceptible. However, the one that wasn't susceptible, that was resistant, there was no Fusarium anyway. There's absolutely none. So it was an interesting trial to have done these and show that they definitely could reduce Fusarium, both the presence of it and then the crop itself, the ability of the plant to grow out and produce a good crop. And I would say this was fine. This is interesting and all that, but what about the possibility that you've been moving Fusarium in that dip tank? And that's one of the concerns that I've had all along. And I finally went ahead and did some trials and found out that if you have clean grow in the dip tank, you don't have anything as far as pathogens. This was a trial that we did a little bit earlier last year. I plated out the dip solution. I'm doing mimicking Janice's trials on the tulips and the daffodils, and plated them out to see what I got out. And this is water. Lots of fungi and lots of weird bacteria. I don't think I've ever seen a blue bacterium before, but that's what we saw here. So that's what the water looked like. This is what the low rate of clean grow looked like. And what we have here basically is one or two fungi growing and then one or two bacteria. And the fact that you have this film all over the plate is from a couple of colonies that just spread because of water. So very, very little. If we go ahead and look at the high rate, we got nobody. We now are killing everything in the tank so that I'm not concerned that a tiny infection in 1 or less than 1% of the bulbs would spread to the others. So we know we're actually killing everything in the tank. So the next thing we're going to do this year is start looking at cuttings and then eventually look at some of the really succulent, perhaps seeds, but probably not. Seed treatment is very hard to make use of and still get the seeds into the flats the way you need to. But at any rate, very, very good control of the surfaces and of the plants themselves when they're infected with Fusarium. Now, it's my opinion, and I can't see any way to get around this, that if you had a vascular wilt Fusarium, if you had it in the vascular system, I don't see how a dip would make any difference to a bulb. However, if they're contaminated from the harvest process and they simply have Fusarium on the outside, then definitely I would expect this to do a great job of controlling them. So let's move on and talk about the fact that some of the little insect pests that we don't appreciate for being insect pests to begin with, they actually manage to move diseases. We've known for a long time that black root rot can be moved, some of the pythiums and phytophthors can be moved, and now we know that we definitely can have Fusarium spores moved on insects. The ones that are the culprits, shore flies, but then also fungus gnats and moth flies. And I'm not an entomologist, and maybe if we have a question about this later, Sarah will help me out, because I definitely don't know enough about entomology to do a great job of interpreting that. But what the University of California researcher did was take Lisianthus, which are just an amazing magnet for Fusarium diseases. There's at least 4 different species of Fusarium on them, and it kills them. This is in a plug that's been put into the ground for flower production. But the disease itself is caused by Fusarium avinaceum. The researchers took stems of Lisianthus that were naturally infected with the Fusarium. So it's not artificially done. It's under normal, natural growing conditions. And what they found was they very clearly proved that shore flies, moth flies, and fungus gnats could become contaminated, as in it would stick to their outside, and that having picked it up simply by walking around on the plant that has the Fusarium, they were able to move it to healthy plants. And this is not the researchers pulling it off of them. This is under natural movement of the insects only. So we know we can move it. They also found it was really odd that it took about 40 days after the flies, any of them, were exposed to the spores for the disease to show up on the Lisianthus. But it only took 25 days if the researchers planted little plants into potting medium that was amended with the spores. So obviously the bugs are not carrying lots of Fusarium, but they're carrying enough to cause the disease. So this is not just a matter of, wow, we found Fusarium on them. This is a matter of it actually moved the disease, and there was that result. Of those 3 flies, even though they all could pick it up, and it was the stickiness, remember I showed you those early pictures of how Fusarium makes spores, that stickiness means that they could get it on them, and then they would fly it all over the place. They did also find that shore flies ate Fusarium spores. So if they eat Fusarium spores, are they going to come through and be alive when they pass through their digestive tract? And they did pass through, they were alive, and they actually transmitted disease that way. So controlling insect vectors on some of these Fusarium diseases may be the key for them to be controlled. And keeping in mind that it's making things even more complex for you, unfortunately, in a Fusarium disease situation. So back to my favorite thing, I like very much to only talk about what works and not spend a whole lot of time about talking about things you can do that really don't help you. And after years and years, I'm going to tell you that Fusarium wilt and nitrogen source and rate, so the type of nitrogen, but the amount of nitrogen, are very intertwined. If you go back over the last 100 and now probably more like 120 years, and you look at all of the research on controlling Fusarium diseases on all crops, not just ornamentals, all crops, then what you find is that certain forms of nitrogen and certain pH situations, whether it is in soil or in potting media, are still the most effective way to reduce Fusarium wilt. The methods in the first half of the 20th century showed that this was the best way to control it and to avoid ammoniacal sources of nitrogen was what the key was for controlling the disease. Now, in the past 100 years, for the most part, we were not growing things in containers. We were using soil, and it was quite a different situation in the field. But the fact is that these crops, and you see here, for some reason, we actually got some work done on an ornamental, but tomatoes, cotton, and banana, huge crops worldwide, that's how they control this disease. So nitrogen, that's important. The effects are very often indirect. And so what it means is you didn't necessarily hand the Fusarium something that it either liked or didn't like, but you caused a reaction in the plant. And so that it's mediated through the plant itself or even through the other organisms in the soil, the microorganisms. So it's not necessarily just because it's a direct effect on the Fusarium. Fusarium wilt on mums, I'll just give you a few bullets on the exact research. They have proved that it is worse as you use higher rates of nitrogen. You can increase it, so you do not ever want to over-fertilize them. Fusarium wilt on hebe and mum, it increased when ammoniacal was used. So not just the rate, but the kind of nitrogen. And Fusarium root rot and wilt is increased, definitely, if you use ammoniacal versus nitrate. It is definitely known to increase if you have lower pH. And part of what you get with ammoniacal nitrogen is a decrease in the pH. Excuse me. So they're all tied very closely together here. You know, I could show you a bunch of trial data, but I don't think it's a whole lot more inspiring than just being told what the summary is. So it's not all nitrogen, however. We also know that Fusarium wilt on some plants, like the tomatoes, cotton, and bananas, actually is worse when phosphorus is high and potassium is low. More experiments showed that if you used, say, the ammoniacal, but you changed the pH, you buffered it, you put liming into the system, and you brought it up to 6.5 to 7.5 in that general range, you could reduce severity. So you have multiple things going on. Some of the studies also indicated that when the pH is higher, the minor elements that are less available are needed for the Fusarium to make spores. So in this case, you're interfering directly with the Fusarium's ability to reproduce by going to a higher pH, whether you use nitrate to avoid low pH or you actually use liming to buffer. And then finally, when we got methyl bromide in the, I think it was the 60s, something around the 1960s, we kind of forgot just about everything you ever learned about how to grow plants and avoid diseases because we could wipe out everything in the soil. And, to some degree, it's good that we don't have it anymore because, frankly, you have to be a better grower if you don't have some of these just amazing biocides to wipe things out. So at this point, and even at that, we knew for a fact that even soil fumigation wasn't as effective as managing the nitrogen, the nitrogen nutrition, and the pH. That was the most important part of managing Fusarium, not whether or not you could wipe out the Fusarium with methyl bromide. And here's an example. How many of you would prefer to use ammoniacal versus calcium nitrate? I'm thinking that this is pretty bad. This is all dead crap. So it's important to see, not just have somebody tell you about it, but to see the benefit. These plants, by the way, were all inoculated with the Fusarium wilt organism from cyclamen. So that is the benefit. It's very important. So can you rescue yourself with a biological or a chemical? I'll show you some of the best products that we have on the market. Some of them are brand new. Some of them you have. Some of them we don't even have in the U.S. quite yet. But there are conventional and biological products or biopesticides that have been looked at pretty consistently. In this particular trial, what we're looking at here, these are the controls. So this is the non-inoculated control, looking really good. And the researcher managed to kill every one of them with the inoculation when they didn't do anything to prevent it. So that's how bad it could look. This is how root shield looked. The plants were bigger and better when they didn't have the Fusarium, but they were alive when root shield was used. You can see that they were okay and they were going to end up making a crop. How about companion? Not as good. We've lost a good, looks like about 50% of them are gone. So it did do some good, but not as good. And here's Soil Guard. Soil Guard was better in this trial than companion, but not as good as root shield. So we can see clearly that the biologicals can help us with Fusarium. It's definitely true. But let me show you a little bit of work that was done on some chemicals. Now these products on the bottom were all from BASF. I could not tell by looking at the handout from Sarah whether or not any of them were registered in Canada, but I don't think they are, at least not under these names, and I couldn't find the AIs either. But let's look at them anyway, because what it turns out is Orchestra is slightly different than Pageant, but on Fusarium it's better. The different colored bars are simply the severity of the disease at different times. Looks like about a week to 10 days apart. You see the untreated ones steadily got worse. Here's Orchestra. It was a lot better, much less disease, but it's still increasing at that low rate. And if you've never thought about why you need to pay attention to rate, this should show you. This is moderate rate, and that's the highest labeled rate. Huge difference here in how well it worked from the low rate to the high rate. And then comparing it to the previous chemical, it really is better at less AI per tank than the Pageant. So when the companies do bring new products, whether they're biologicals or not, if they're telling you they're better, then at least assume that that's possible and check it out for yourself. Another trial, this is the Fusarium wilt on basil. A lot of these plants are not registered. In fact, I don't think any of these, a lot of these aren't even registered in the US on basil. But it's an example of some of the trial work that we have done by IR4 in the U.S. to try and help registrations on minor crops. And a lot of things are done for research sake and that we wouldn't say are ready to be used yet. In this case, this is the rating of how good the plant was. So a rating of 5 is just 100% healthy and looks great. That's non-inoculated. When they were inoculated, they were just about wiped out. So that low rating shows very poor plant health and a lot of Fusarium wilt. Root shield did not work in this particular trial. Palladium at different rates, Tourney and Trinity. They work somewhat. The Trinity worked the best at the high rate. These products are not registered as drenches. Palladium is. These are not. And they're not in a group that we even have registrations for Fusarium, at least a wilt situation. But the one that you do have, 3336, is thiophthanic methyl. And it may not be identical to Senator, but it is definitely the same active ingredient. And this was 100% effective. Thiophthanic methyl is systemic. It is known to move in plants and has been one of the only things that we have for years thought in a conventional system was going to give us some good results on Fusarium wilt. And you can see that it definitely did here. And there's another product that is, I can't remember yet whether Torque is registered for drenching, but it is definitely not a Canadian product at this point. But the point is, the one thing you do have really did do a very good job. And I'll show you another. A lot of the same things you see here, kind of, this is the again, the same IR4 funding system. A lot of those same plants or rather same products have testing. This is on a stem rot, so it's not a wilt, a straight stem rot. And this was done at University of Florida by Aaron Palmatier. The ones that I put in the green banding are the best products. 69% disease severity on the inoculated ones. The Root Shield Plus did a very good job. The Heritage, and again, the 3336, or if you think you do have Heritage registered, so you see that, and then 3336 just did a fantastic job. The fact that they all have Ds, these guys I've got in green, showed they're all statistically the same as each other and the same as if you had not even infected the plant with the Fusarium. So you have all three of these registered in one form or another in Canada. I know you have Heritage and Root Shield Plus definitely, and then Senator would be at least roughly equivalent to 3336. This is a trial we did last year. I like cacti and succulents, especially like Christmas cacti. And we did a trial on the Fusarium stem rot. You can see it causes a terrible collapse of the plant. And it's this is one of the things that happens. The tops just pop right off because of this infection at the base. What I'm showing you here, a rating of 5, number of spots. So we had a maximum of 5 spots per plant for the inoculated. The Heritage did a little bit, but not that good. Orchestra was the best. So these chemicals all have strobilurins in them, and they are not equal. So again, just to give you a little heads up, if a new strobilurin comes to your registration process, it is possible that it may work better on certain diseases than the ones you already have. We did a trial last year also, a couple trials actually, on Fusarium wilt on cyclamen. This shows you the treatments that were in the trials. We had Heritage again. Apopka is a new biocontrol agent that's just been registered in the U.S. and comparing it to Root Shield Plus. You can see the rates of the drenches, that's fine. You can also see the intervals, and I will ask you to keep in mind that the Root Shield Plus was used one time in this trial, one time. And the Apopka, which is another biocontrol, and Heritage, which is conventional, were used every 2 weeks. So here's what we saw happen. Again, this is the inoculated. In this particular case, a rating of 5 is every plant is dead. Heritage did a pretty darn good job. Mural did better. Mural has some heritage in it. But then we have a bunch of, worked a little bit. But let's look at the plants themselves. The non-inoculated plants, thankfully, stayed healthy, which is always happy for me. The inoculated ones, we did a pretty good job of killing a bunch of them. We have a few that didn't really seem to get too many symptoms at that point. Here's what Mural looked like. Remember, that is our best treatment up here in terms of how bad the disease was. And we had one dead plant and then some that were kind of poor quality for sure, but we had some very good looking ones. And then here is the new biocontrol. And while this is not as good as this biocontrol, not as good as the conventional, it's certainly better, somewhat better than the inoculated controls. I'll show you another trial that we did. This is a separate trial, but in this case, we had a few other things in it, all conventionals in this case. And this is the percent mortality. So of the 10 plants, there's 10 pots, which we arranged in a randomized complete block here on the side. Of the 10 of them, at the first rating, one of the non-inoculated ones was dead and two of the inoculated ones. So this is 10% or 20%. One of the heritage was dead. There were no dead ones in Mural. Broad form, back to that strobilurin combination product. One of the 10 dead, two of the 10, but let's see, that's not all there is to it. You have to do the trial long enough. The disease just kept going. By the end of the trial, we had lost 40% of the inoculated ones and 20% of the heritage treated. So you really have to make sure that you look at the long enough trial data. One thing I want you to realize, I do not have any better Fusarium product to try than heritage, and it very often is not very good. It could be really good compared to doing nothing, but not up to speed like you might get with a pythium product or a phytophthora product. So I'm getting to the point where I'm just going to show you an overall summary. I recently went to the trouble to try and find everything out I could about all of these biocontrol agents and a great many more. And actually, some of these are biologicals and some are biopesticides. So we have a big range of things, but they're in that category. And I summarized them across all crops. So every piece of data I could find, whether it was on spinach in a field trial or cyclamen in a potted trial, I've summarized them. And the two products that have the best shot of giving you good results are Pre-Stop, which you do have registered, and Root Shield Plus. They are the very best for Fusarium. Now, mind you, I have summarized them across all crops, all diseases. So this isn't the only story, but since we're talking about Fusarium today, this is it. If you wish to be or have to be in the biocontrol or OMRI or organic category, then these are the two that I would focus on the best. You can see the range is quite high, but that's because, again, all crops under all types of growing conditions, if you start summarizing them, you can definitely come up with a trial where you had no control, but other trials where it was good. So it's got a really big range of activities. And this is not unusual. This is not any worse for biocontrol products than it is for conventional products. They often give you a very big range, but you do see surprising consistency with Pre-Stop and also up to very good control with Root Shield Plus. So that's my recommendation for you if you're fighting Fusarium and you need to be in the organic range. What about all products? I have summarized most of this across just ornamentals, but also adding some information from other crops here and there. These are the best products for Fusarium. Medallion, you have registered. It's in a FREC group 12. This is a formulation that you don't have, and I don't believe you have Palladium, which has 12 in it plus another one. So we're looking at Medallion, Heritage, plus all these other things I showed you in the other trials. Where we got some control of Fusarium, but you don't have them. So you got Heritage, TerraGuard, also a very good product for Fusarium. Thiophenic methyl, that's Senator again, and then Root Shield Plus and Pre-Stop. This is it. If you insist on doing treatments for Fusarium instead of fertility and sanitation, this is what you have. You can definitely do some rotations. The only thing you don't want to rotate, and since you don't have it, you're good. You do not want to rotate TerraGuard and Root Shield Plus, because TerraGuard is one of the things that will kill Root Shield Plus. So that's a pretty disastrous thing for you if you did both of those. So that gives you the overall summary. This is just like bottom line. If you wish to be using products instead of cultural type controls that can be much more effective on Fusarium, then these are your best choices. So just as a review in general, you've got to know whether you have a Fusarium problem or not, which means you have to look at your plants and you have to throw the stuff out if it looks like it has it. You've got labs up there, get them diagnosed one way or another. So you've got to be really on top of scouting and roguing. You need to manage insects in certain situations, having proven that we can see insects like shore flies, moth flies, and fungus gnats move Fusarium spores, and not just move it around how, researchers entertain because they can show it. It's moved the disease. Now you have spread disease problems. That's a real possibility. You have to really think about other sanitation type thoughts. Am I starting with bulbs that might have it? Am I cleaning the benches or the floors or whatever surface you're dealing with in between crops? You've got to do that to get rid of as much of the inoculum as possible. And again, probably most importantly, get away from ammoniacal nitrogen. Get away from low pH. If you can get your pH up closer to 6.5 to 7 or so, you're going to reduce the severity of Fusarium so that it's not necessarily a matter of trying to wipe it out, but live with it, but it's unhappy and it's not going to cause disease. And then finally, again, if you insist on it, I've given you the short list of what works in the biological and conventional product category. You can definitely do that, but I will tell you after doing Fusarium trials for almost 40 years that if I had to do this, I'd be paying attention to this, the first 4. I would not think I had any hope in the world of fixing things with any of these products. If you can do these other things and then perhaps use a biological to change the fertility situation, because they do interfere also, and interfere is probably not the right way to put it, but they interact. Anything you can do to help the health of the plant in a Fusarium situation, you're going to be better, because reducing stress is one of the key elements of keeping your losses from Fusarium to a minimum. So I think I've said enough, and if any of you have typed in any questions, maybe Sarah will let me know what they are, or if she has any, that would be great too.
Okay, well, thank you, Anne, for that. We've run out of time a little bit in terms of questions. So what we're going to do is we'll send Anne the questions afterwards, and we'll post the questions and the answers on Greenhouse Canada online. Thank you for the wonderful talk today. As a reminder to the audience, a copy of Anne's slides are available in the handout section to your right. A recording of this webinar will also be made available on our website and sent out after 24 hours to everybody who's registered. For more great content on greenhouse ornamental production, visit onfloriculture.wordpress.com. That is a blog that is regularly updated by Sarah and her colleagues at OMAFRA. And of course, check out greenhousecanada.com for news and production tips. Be sure to register for our 3rd webinar in this series. It's not going to be next week. It's in 2 weeks on April 2nd. We'll be talking disease diagnostics with Dr. Karen Rain of the University of Maryland. See you next time.